Today we will be talking about a study on antibiotic resistance by Zhao et al.
While natural selection is the main mechanism by which bacteria populations resistant to antibiotics develop, the resistant genes are actually a product of random mutation that occurred during replication. And as we continue to use antibiotics worldwide, bacteria without a resistant gene are selected against. Let's take a look at this concept in a different way. Say two friends, one short and one tall, are out on a casual jog when suddenly a vicious tiger escapes from the nearby Atlanta Zoo. For the sake of the example, let's say that the person with the long legs is able to outrun the person with the short legs, leaving them behind to deal with the vicious tiger. The person that escaped is able to live on and potentially fall in love with an ideal mate to make long-legged babies. In this example, the escaped tiger is the antibiotic, and the two friends are the bacteria. The friend with the long legs is similar to a bacteria with an antibiotic resistance gene. And once the non-resistant bacteria is eliminated, the resistant bacteria would no longer have to compete for resources to reproduce, giving rise to a more resistant population.
Let's look at a study done in 2016 by a group of Chinese scientists who observed antibiotic resistance genes, or ARGs, present in estuaries along the Chinese coastline. Aquatic environments are essential for life in China, so the health of these coastal environments is crucial. In recent years, about 60,000 tons of antibiotics have been released into Chinese water streams. Prior studies have shown the existence of ARGs in estuaries. However, a more comprehensive study was needed in order to identify all of the driving factors contributing to ARGs, including environmental factors, geography, human factors, and pharmaceuticals. The scientists hypothesized that many different types of ARGs would be found in their sample sites due to the variety of these driving factors present in these aquatic environments. In order to conduct this study, the researchers took sediment samples from 18 different estuaries along the Chinese coast and analyzed their DNA and chemical content. HT-qPCR techniques, which are basically reactions that single out certain DNA sequences by copying them over and over, were used to determine the abundance and diversity of ARGs. Environmental factors were determined by taking the pH of the soil and calculating the carbon and nitrogen content through combustion. The socioeconomic or anthropogenic factors such as GDP, sewage, total population, meat production, total wastewater, and number of patients diagnosed and treated were taken from official records and databases.
Statistical tests done on a wide variety of socioeconomic factors and the abundance of ARGs in the estuaries showed a significant correlation between population size, GDP, and sewage produced with abundance of ARGs. Another statistical test called PRDA was used to distinguish the effects from anthropogenic, environmental, and bacterial community factors, and it showed anthropogenic factors had the largest effect, while bacterial community, previously thought to be a significant factor, had the lowest contribution to ARG abundance. The results from analyzing the 90 sediment samples showed that the proportions of the different types of antibiotic-resistant genes, or ARGs, in each estuary were relatively similar. Although 248 different ARGs were detected in total, only 18 ARGs were found in all 90 samples. Multi-drug-resistant ARGs are the most common type of resistant gene, likely due to the wide variety of antibiotics used today. After detecting the types of ARGs in each sample, the samples were tested for any antibiotic residue. These tests show that the estuaries with large amounts of tetracycline residue were significantly correlated with high amounts of each type of ARG tested. Macrolide residues were the only other type of residue significantly correlated with high amounts of any type of the ARGs. An article by Stuart Levy details how antibiotic-resistant genes can quickly travel from bacterium to bacterium through the transfer of plasmids, resulting in a swift rise of ARGs.
The study found that although selection for ARGs can be driven by environmental factors and interactions between different bacterial species, selection is mostly driven by human and livestock activity. This becomes a huge issue as China's population and economy continue to grow, increasing both the quantity and quality of antibiotics released in waste streams. More antibiotics in the water lead to more antibiotic-resistant genes developing, and more antibiotic-resistant genes mean stronger, more resilient bacteria that can cause deadlier infections. And what of our fine furry friends and their long and short-legged prey? If the zookeepers continue to be negligent of proper safety precautions, vicious tigers will continue to escape into the general population to snack on unsuspecting short-legged civilians. And as the escaped tigers continue to select against short legs, the surviving population would be prepared to outrun any predator released from the local zoos. Their increased longevity would allow them to reproduce and repopulate, giving rise to a physically and evolutionary fit race ready to dominate the world.
While there are many solutions available to halt the issue of more ARGs arising and affecting human and animal health, there are a few that we will focus on. One solution would be to eliminate the resistant bacteria by introducing more non-resistant bacteria that would serve as competition to the resistant type. This way, the resistant bacteria would become less abundant as it competes with and is lessened by the non-resistant bacteria. Another solution would be to reduce the use of antibiotics and to limit the exposure of these antibiotics to the environment. By overusing antibiotics, more bacteria become resistant to the medication, therefore eliminating any effect the medication may have on preventing diseases in humans and animals. With a less frequent use of antibiotics, these bacteria would not build up such a resistance to the medication, thereby allowing the antibiotics to serve their purpose of disease prevention and treatment. Also, regulation of hospital waste disposal containing antibiotics could be largely increased.